Okay, that was row-dependent termination. Let's now move on to row-independent termination. Basically, there's no row protein here. What ends up happening is the gene sequence itself actually has a region with lots and lots of uracils. Instead of like a poly A, you have a poly U sequence. And so that's what this picture is showing you here. As the RNA polymerase kind of goes along the sequence here, you'll have a uracil-rich region in the sequence. And what that does is it's adenine, but it produces a uracil-rich sequence in this region here. And we have this other protein called NUS-A. Oops, wrong thing. I was hoping that I had my highlighter there. So NUS-A is going to cause like a pause so that once enough uracils are made, you have basically, if you guys remember back to base pairing, C's and G's have 3 hydrogen bonds, all right? 3 hydrogen bonds, while hydrogen bonds aren't very strong, that's still stronger than 2 hydrogen bonds that you get between AT or AU. So that NUS-A helps put pressure on the RNA polymerase along with this loop stem that also occurs in the sequence, and that causes the new RNA to peel off. So that's the big difference between row-dependent and row-independent, is the row factor in row-dependent that chases along that loop and stem and then causes pressure to push the two apart. And in row-independent, that NUS-A and that loop stem with the numerous uracils present that cause dissociation of the RNA polymerase from the RNA strand. And then the double helix glues back together again. So that's transcription in prokaryotes. Remember, in prokaryotic cells, what do these guys have? No nucleus. So DNA is just kind of like a bunch of spaghetti inside of the cell. And because of that, the RNA can immediately be translated. In eukaryotes, things are a little bit different because eukaryotic cells, as you all will remember, have what? Eukaryotic cells have the nucleus. So the RNA actually has to leave the nuclear space. Now, one of the things to keep in mind about the nucleus, just as a review, let's try to draw a big nucleus, okay? The nucleus itself has a lot of little, thought that might work, has a lot of little spaces in it, a lot of little, what we call nuclear pores. The nuclear pores are what I consider the nuclear doors in and out of the nucleus. So here are your nuclear pores, okay? And stuff can go in or come out. And in fact, this is how RNA gets out, and this is how transcription factors get in. They travel through the nuclear pores, but there's no transport directly across the nuclear envelope. So this red area here for the nucleus, that's not something that things can travel through easily. If it's a hormone, that's a little different, and that's physiology, that's a different class. So when we look at transcription in eukaryotes, one of the things you need to bear in mind is that the cells are larger. They're much more complex. They have membrane-bound organelles, including the nucleus here. And so the increased complexity also is reflected in more genes. Now, don't fool yourself. The organism that has the most DNA on the planet, the highest DNA content, is a tiger lily. So gene number and DNA content are not linked with complexity of the organism. Having said that, multicellularity does seem to go along with having more genes and having more highly regulated genes. So nuclear DNA is transcribed by 3 different RNA polymerases in eukaryotic cells. You have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now, why am I designating nuclear DNA? Do you guys remember back when we were talking about extrachromosomal gene transmission? Remember that? With mitochondria and chloroplasts? Remember, they have their own DNA molecules. So we're not going to deal with how those are replicated. Right now, we're simply dealing with oops, trying to figure out what I did there. Right now we're just dealing with nuclear DNA that you find in the nucleus. RNA polymerase 1 itself transcribes all ribosomal RNA genes. And so what we say is that they make basically all the different types of RNAs except for the 5S ribosomal RNA. Don't worry about that right now. That's an upper, I guess that would be like the next genetics class. RNA polymerase 2 makes messenger RNAs and some snRNAs, okay? So basically when we're talking about RNA polymerase making messenger RNA, this is what we're talking about, RNA polymerase 2. RNA polymerase 3 makes transfer RNAs, right? So the way I remembered it was rRNA is number 1, mRNA number 2, and then tRNA number 3, okay, or RNA polymerase 3. Now, yes, you can make some ribosomal RNA here, but for the most part, in eukaryotic cells, RNA polymerase 1 makes ribosomal RNA. And it turns out that most of these RNA polymerases are very similar to one another. You don't need to worry about the details, but here is an example of eukaryotic RNA polymerase 2 from yeast. It actually looks even similar to bacterial RNA polymerase that makes messenger RNA. Okay, there is some overlap with ribosomal RNA, tRNA in prokaryotic cells, but don't worry about that right now. So, what are we dealing with here with the eukaryotic structural genes? Remember, structural genes produce proteins that are used in the metabolism of the organism. So, the structural genes are going to contain also regulatory elements. That would be like your -10, well, more of the promoter, regulatory elements within the promoter. The -10 would be the core promoter or the TATA box. Again, we're back to the TATA box, right? That's sitting next to transcriptional start. And you also have some new items called enhancers and silencers. Enhancers change the speed at which messenger RNA is made. Not all genes express messenger RNA at the same speed, which is kind of weird, right? Some genes you need to, or some proteins need to be synthesized very quickly. If you think of things like, oh, I don't know, anything that would be involved in glycolysis. You need a whole lot of enzymes to turn glucose into pyruvic acid. Those particular genes would have enhancers to ensure that those enzymes are being pumped out constantly so that the cell can make energy from glucose. So in this picture here, what we're looking at are the regulatory elements that are far upstream of our -10 and -35 consensus sequences. And in fact, here in our eukaryotic cells, you'll find the TATA box at -25. It's kind of in a different space. Here, the regulatory elements will be found between -150 and -100. And again, these are going to be your enhancers and silencers. Why would a gene have a silencer? You don't need to worry about that right now, but sometimes genes have regulatory elements that allow them to be expressed only under specific conditions. When that condition is not met, the silencer is active. So, what do we have to control gene expression based on where the gene is located? So, for example, which chromosome the gene is located. We have one group of elements called cis-acting elements. These are like the TATA box, enhancers and silencers. What these do is they directly exert control, okay, over that gene. They are basically turning the gene on and off, all right? But it's only over a given gene at a time, not the whole genome. Transacting elements, these are crazy because these things actually regulate the cis-acting elements. So, cis, the way I remember it, controls the gene, controls gene expression. Trans controls regulatory proteins or elements. So I'll say reg, pro, or elements. So for example, if you have, let's come back up here for a minute. If you have a silencer, a trans element would be controlling whether or not that silencer is working. Fun, huh? All right, so let's start with what RNA polymerase II does with a term that I keep saying over and over again, transcription factors. Transcription factors help RNA polymerase stabilize. So when I was showing you like these bottles of ink stacked on my wrist and on my hand, you know, this is not easy actually. This one right here especially likes to wobble around. And then I put brain over here in the middle to be sigma factor and so forth. If you want these things to stick and stay together or to really hang in a spot, it helps if you have proteins that help stabilize these things. Now, why is that important? Something that we all fail to remember, and I think rightfully so, is that most of our cells, or at least human cells or mammalian cells, are living at somewhere around 98.6 degrees, okay? Stick your finger in a bottle or a pan of water that's at 98.6 degrees, right? At human body temperature. That's hot, all right? That's a cool jacuzzi. That's a bath. And so all of our molecules are sitting at this relatively warm temperature. What is heat? Heat is kinetic energy, energy of movement. So the warmer the environment, the more that these molecules are going to be vibrating around, including DNA, including RNA polymerase molecules, and including the subunits that need to assemble. So these things are shaking around like maracas, right? But they really need to be stable. That is part of what transcription factors do. They stabilize them into place. So there are several different elements that come together for stable transcription to happen. First of all, of course, our RNA polymerase II, which makes messenger RNA. There are general transcription factors, which we'll get to later, and then there's another protein called the mediator. Oh my gosh, can't get more complicated. Actually, yes, but we're not going there. So here's an example of transcription factor D binding to the TATA box here. All right, so this is all supposed to be upstream of a gene. I really wish that they would have identified where the gene was here. Let me get a color. Let's make the gene orange here, okay? If transcription factor 2D and transcription factor 2B bind, then you'll have RNA and then transcription factor 2F. When these all come together with RNA polymerase II, you have stabilization and initiation, or at least the ability for initiation to start. What happens is that these elements come together. So remember when we were looking at alpha, beta, sigma in our prokaryotic system, eukaryotic cells have similar proteins, but they don't make the same type of round shape of a whole enzyme. So this is what we call the pre-initiation complex. It's a closed complex, meaning that these proteins come together and kind of wrap around. So transcription factor 2D, 2B, 2F, 2H, and 2E. And these all come together to allow transcription to start. There are some other things that happen for transcription to initiate, but once RNA polymerase starts going down the chain of DNA, these transcription factors release. All right, so basically what happens is you have them floating around in the nucleus and they assemble, okay, and I'll use the same little goofy things I have here. Throw an eraser in here, right? They all assemble to make a holoenzyme, and then we're going to now make a piece of RNA, messenger RNA. Several of these come apart. Transcription factor 2F maintains its contact with RNA polymerase II, and then ATP is being used to synthesize new DNA, or new RNA, I'm sorry. So what we call the basal transcription apparatus is going to be RNA polymerase II and the 5 transcription factors, okay? So basically, this picture is going to be pretty important for the exam. We will finish up with termination in the last video.